Hello, my name is Alaa Aziz, and our research focused on the enzyme F420-dependent glucose 6-phosphate dehydrogenase, which is found in Mycobacterium tuberculosis. This enzyme is responsible for the activation of prodrug, which treats multiple drug-resistant and extreme drug-resistant cases of tuberculosis disease. The FGD crystal structure was resolved by our collaborator, Dr. Ghadar Bashiri. It is a 78 kilodalton homodimer. This is an expanded view of the active site, where we see that three important amino acids are believed to play an important role in catalysis. Those are glutamate 109, histidine 40, and tryptophan 44. This is a proposed glucose 6-phosphate binding site where we believe that glucose 6-phosphate would bind. The F420 cofactor is spectrophotometrically distinct. The oxidized form shows a high absorbance at 420 nanometer. Upon reduction, it loses its high absorbance and gains a shoulder at 320 nanometer. This is what allows us to do the type of chemistry we do in our lab, where we look at the reduction rate of the F420 cofactor. FGD catalyzes the conversion of glucose 6-phosphate to 6-phosphogluconoloctone. It facilitates the transfer of a hydride from carbon 1 position of glucose 6-phosphate to the carbon 5 position of the cofactor, reducing the cofactor. Dr. Mesu Uyugi was able to determine the order of substrate addition. She determined that F420 binds first, followed by glucose 6-phosphate. Using pH profile, she determined that glutamate 109 is acting as an active site acid and donate a proton to the N1 position in the cofactor. It becomes protonated. She was not able to determine the active site base that abstract a proton and facilitate the hydrogen transfer to the carbon 5 position of the cofactor, reducing the cofactor and producing 6-phosphogluconoloctone. This is when I picked up the project, and my goal is to answer which active site amino acids serve as the active site base. I expanded my studies on additional amino acids present in the active site, and those are histidine 260 and glutamate 13. We created some variants to study the role in catalysis, and I also continued my studies on the H40 amino acid. Our binding studies showed that these amino acids are involved in F420 binding, but they are not involved in glucose 6-phosphate binding. And this is shown by the significant increase in binding constant compared to wild type in the case of F420 binding, but not of much of a difference in the KD in the case of glucose 6-phosphate binding. Our steady state kinetics showed that these these amino acids are involved in catalysis. This is shown by the significant decrease in KCAT and also catalytic efficiency compared to wild type. Our pre-steady state experiment that we do on the Stoffel spectrophotometer that collect data on a millisecond time scale, and then we get the data, we fit it to a model that we can create on Kintech Explorer. In this model, we know that cofactor binds first, followed by substrate. I took into account the possibility of an intermediate turning over in the reaction and also product release. We learned two things in this from this experiment. That is the absence of intermediate. We do not have any intermediate. This is shown by the complete overlap of the blue line, which signifies the product release, and the green line, which signifies the intermediate that I took into account. And we also have a fast chemistry slow product release reaction. Our pH-dependent studies showed that neither of these amino acids are serving as the active site base. However, glutamate 13 might be assisting somehow in the reaction. This is shown by the wider bell-shaped curve compared to wild type. I turn my attention to the NADP-dependent glucose 6-phosphate dehydrogenase mechanism. And in this mechanism, they have histidine 240 and aspartate 177 forming a catalytic dyad. Histidine 240 acting as an acid donates a proton to aspartate 177 and then acting as a base, abstracting proton and facilitating the transfer of a hydride to the NADP. So this got me to think of the role of our glutamate 13 in this reaction. In our reaction, I came across a server called ProPKA, where you are able to upload your crystal structure, and it will predict the pKa of each amino acid present in your enzyme. This, along with our pH profile, suggests that histidine 240 and glutamate 13 are forming a catalytic dyad. Histidine 40 is acting as an acid first, donating a proton to glutamate 13, and then acting as a base, abstracting proton from glucose 6-phosphate and facilitating the hydride transfer. So I would like to thank my advisor, Dr. Johnson Winter, along with current and previous lab members.